This is Stanford Engineering's The Future of Everything, and I'm your host, Russ Altman. Today, Professor Ali Boehm will tell us how she's using wastewater to monitor the COVID pandemic. Yes, COVID makes it into the wastewater, and you can measure it. It's the future of wastewater epidemiology. We don't usually think about wastewater very much. You flush the toilet, you put stuff down the sink, and that's it. But as you know, if you think about it, all of that wastewater is collected into bigger and bigger sewer pipe mains that wind up in a sewage plant. And that sewage plant then processes the water for safety and sends it wherever it sends it. Well, it turns out that when we flush things down the toilet or put things down the sink, it contains a lot of our bodily fluids, which can include the viruses and bacteria that we are infected with. So it turns out that you can actually monitor public health issues like the pandemic of COVID by looking at the wastewater and measuring how much COVID you see from a certain region. As long as you know who's contributing to the sewer pipe, you can figure out how much COVID is in that population. This has led to the idea that you can actually do epidemiology, the study of disease in a population, by monitoring wastewater. You could do it for a bunch of diseases, you can do it for animal diseases, and really, perhaps, the opportunities are endless. Well, today, Professor Ali Boehm from Stanford University, she's a professor of civil and environmental engineering, as well as a professor of oceans at Stanford, will tell us how her group innovated to create wastewater monitoring in the COVID pandemic. Ali, to start off with, can you tell us how wastewater-based surveillance of infectious disease generally works?
Yeah, well, anything that you put down the toilet, the drain, or a drain in your sink or the shower, it ends up going to the wastewater treatment plant. And so that sample going into the wastewater treatment plant is just like a large biological composite sample. And so if you get that sample, you can use it to probe which infectious diseases are circulating in the entire contributing population to that sewage treatment plant. So that's generally how it works.
Great. So I have so many questions because this is like a surprising and exciting thing. So the first question is, how do you know, first of all, how do you get to that water? And how do you know who's contributing, so to speak, to whatever the fluid is that you sample and test?
Well, the way that we get the sample is we have to work with the wastewater treatment plant. So this project, our research on this relies on the plants cooperating with us and providing us samples from their facilities. So that's how we get the sample. And most of the facilities have, they're large engineering unit processes. So there's lots of ways to sample at different locations throughout the facility to make sure it's operating. So these are samples that they're already collecting for other purposes. And so we can, just piggyback on what they're doing to get the sample. And then in terms of how do we know who's contributing, most wastewater treatment plants have a map of their sewer shed, and they know where all the pipes are coming from that lead to the plant. But oddly, some older plants don't have these necessarily. So they have to do a little bit of work to figure out where their so-called sewer sheds are. But that's how we figure out who's contributing to the sewage.
So is all of the sampling done at the plant? Because I'm imagining a tree of contributing like homes and buildings that then they go into a main, which goes into a bigger main. So do you have the ability? How much can you go to a single dwelling or an apartment building or a neighborhood, or are you really limited by everything that comes into that single huge plant?
So most of our work has been focused at the single huge plant, but you're right. You can go up the sewer shed, as we call it, or climb the tree that you described, and you can get samples at different junctions. So we have done some work at Stanford University on campus, sampling adjacent to dorms. We sample a sub-sewer shed of the university at like a large sewer main where we put in a composite sampler to get the samples out. We pump it out of the main. And we've also worked at some K through 12 schools where we've sampled like little campuses. So you can go all the way back to the household, but I think the question is like, what is your objective with
Right.
the project, and like, does it make sense? Why not just give everyone like a test, like an individual test, if you're going to sample
Right.
at the household?
Okay, great.
Yeah.
So that was just the background. I just wanted to get a feeling for like the fluid flow and where you're sampling. But let's go back to like first principles now is what are the diseases that you find most critical to test for? And tell me a little bit about how that's going and what have we learned from these testings?
Yeah, I mean, there's a lot wrapped into that question. So I think the question of like, I think there's two parts of that question. One is like, what does this technology work for? And then the second question is like, how do you choose what to monitor for? What's going to be most useful? And so first I'll talk about like the answer to the first
Great.
question. So amazingly, we are finding that wastewater based epidemiology works for almost every disease that we've studied. So we started off with COVID-19, and we found that what we were detecting in wastewater matched amazingly, unusually well with reported cases. And then we started tracking different variants, and we found that we were able to detect different variants, and it also matched like so well with what was in the case data. And then we expanded our work to influenza A and RSV, respiratory syncytial virus. And we found also that the concentrations of RNA from those viruses in wastewater matched really well with both passive and active surveillance data done in clinical laboratories. And then we expanded our work to mpox and amazingly found
as in monkeypox.
Yes,
Okay.
which is now we say mpox.
We have a new name for it. Thank you.
Yes. So mpox concentrations of DNA match amazingly well with reported case data, and we have worked closely with CDPH, California Department of Public Health, to confirm that. So it works for mpox. And then we're just finishing up putting together a paper where we looked at para-influenza, metapneumovirus, influenza A, influenza B, RSVA, RSVB, seasonal human coronaviruses, and what have I forgotten? I think that's it.
Wow.
Oh, right, human rhinovirus. And it works amazingly well for all those as well. So like, I'm really amazed that it seems to work for everything we've tested, and our focus has really been on respiratory viruses, respiratory diseases, etiologies of respiratory disease, and then mpox. And so we haven't worked on other types of viruses or diseases or bacteria yet, but we will. So far it seems like it works well for everything, like, which never happens in science.
Yeah.
So it's pretty surprising.
So when you say it works, and you said the magic letters of DNA, and you might have said RNA as well. What is the actual kind of assay or measurement that you make?
Yeah,
how hard is it to do, and how expensive is it, I guess?
Yeah, so we're using RT-PCR and PCR in a digital droplet format to get concentrations. And in terms of how hard is it, I mean, we do have to do some assay design, which is pretty easy nowadays with different programs and like all the information available regarding the genomes of these organisms. I think our biggest challenge is evolution of the viral genomes, which unfortunately occurs.
Right.
And then the question of how expensive it is, I mean, I think, compared to a banana, it's expensive, but if you can get one sample and know what's happening in 1 million people with one assay, that's way cheaper than testing 1 million people. Right. So the costs of it are very similar, I would say, to like getting a clinical test. But the question is, like, how much does a clinical test cost? Because we have insurance and there's different costs for different insurance carriers. So that cost is sort of like unclear. So
it's very squishy.
but it's not expensive. I mean, Yeah.
Now, when you make these tests, I think you refer to this also in your comments. I'm wondering how quantitative it is. Like, if you go there Monday and you get a measurement of 100, I'm making up these numbers, obviously, and then you go there Wednesday and you get a measurement of 300, is that interpretable in terms of there's been an increase in the population? I'm just wondering what causes, because it's kind of amazing that this fluid, which is literally waste, and which you might think is such an inhospitable environment that these viruses might not even survive. But obviously, you've already shown that you're reliably seeing their DNA and or RNA, which is these are the for those of you who don't think about DNA and RNA all the time, these are the genomes that encode for the life processes of the virus or the bacteria. And we have genomes as well, we humans. So there are fingerprints for these organisms. So my question is, how quantitative is it, and how interpretable are those numbers in terms of what's going on in the population?
Yeah, well, I'm an engineer by training, so I'm very quantitative, and our concentrations are very quantitative, and we have a measurement with errors associated with it, and we run 10 replicates. And so we have a really good sense of the quantitation. And I think what you're referring to is variability. So, how do you discern true increases from just like stochastic variability in the
Right.
concentrations? And I think, one thing that we have tried really hard to do is to introduce our public health and medicine colleagues to the concepts of variability in the environment. So they are used to seeing variability in like syndromic data and clinical specimen data, and they know how to interpret that. Like, oh, the levels are low on the weekend. That's because nobody does tests on the weekend. So they're comfortable with that. But for environmental samples, you're always going to have variability that they are perhaps not accustomed to. And so to get to your original question, how do you know if like the level on Wednesday is higher or lower than the level on Monday? I think we have been exploring different approaches to identifying trends in the data. So that's something that that's like the crux of the utility of this for like public health protection and pandemic response, is knowing when an increase is a real increase
Right.
versus stochastic variability.
Right.
Yeah.
So you mentioned your partners. How are the public health organizations receiving this? So I'm imagining this is a little bit disruptive to the way they normally like to do surveillance. And even the best people don't always love to be disrupted in their professional lives because it means change. And so how do you approach them? How do you explain the possibilities and potentials of this? And how do they receive it when you go to your public health colleagues?
I will be honest and say that in the very beginning of the pandemic, we could not get anyone in public health to really return our emails or phone calls for obvious reasons.
Right.
And the first group to really engage with us was Santa Clara County. And Dr. Sarah Cody assigned someone basically to interact with us, and our relationship grew. And then eventually, CDPH at the state level,
So that's the
they
California Department of Public Health.
right. They were able to engage, and we now there's actually someone working there who was a fellow at Stanford, and so they've been really great, amazing partners. And I was just reflecting recently on how our relationship has grown and changed over the last 2 years. Where at first we were really trying to convince them and show them and teach them about this approach. And now they are like huge advocates for this, and it's part of their plan for the future for the state of California. So it's been really amazing to see this like uptake of this new technology at the county level and the state level where we are working. We also worked really closely with CDC in the beginning of the pandemic. Actually, a former student was working on this at CDC, actually two of my former students. So I felt really lucky that we had that connection. Those two have since cycled off the wastewater surveillance system at CDC, and there are new people there that I don't work as closely with anymore. But CDC has really embraced this technology as well and see it as something that will be sustained into the future as well.
What is it about these measurements that gets them excited? When you talk to them about this, my guess is it's filling a blank that they had. What is the blank, and how will it kind of improve their ability to make public health decisions or interventions?
Well, I think the blank is that the test seeking behavior and testing availability to the general public is really variable. So there's always a concern that the people getting tested for different diseases represent just an odd group, the tip of the iceberg. You never know if what you're seeing in the like surveillance data is actually reflective of what's happening in the community. And wastewater, on the other hand, is totally unbiased. It doesn't require any behavior change. It includes everybody, so it gives you this sense broadly of what's circulating in the community.
Great. So the there's so many directions that I could go, but one of the things I wanted to ask is one of the things that people talk about is cleaning wastewater so that we can recycle it and reuse it. And the knowledge now that our wastewater is filled with these pathogens might make some people pause about their comfort with recycled water. So what is the way to think about that? And how should people be worried that your findings are actually indicating that recycling water may not be a good idea, or how should it be interpreted?
Well, first, the measurements we are making are of genomes of these viruses, and they're very short segments of the nucleic acids. And so they're very persistent in wastewater. We've done studies where little segments of RNA are persistent for weeks. So we're not detecting live virus. But you should know that there's a lot of nasty stuff in sewage, and it's always been nasty stuff. I mean, it's poop, it's vomit, it's urine, it's blood, it's mucus, it's sputum. I mean, that's always been there. And so when we're sampling wastewater before it goes into the treatment plant. Treatment plants have been designed to remove bacteria and viruses. And so those unit processes are still working. Wastewater has not changed. And we know that those unit processes make wastewater safe when it leaves the wastewater treatment plant. And then if it's going to be used for water recycling, it goes through a whole bunch of other unit processes that continue to remove things. So I don't think that our work has any implications on on the usability of wastewater effluent for water reuse.
And not to mention the fact that you certainly could check all the effluents and make sure that they're clean to whatever standard the authorities want. This is The Future of Everything with Russ Altman. More with Professor Ali Boehm next. Welcome back to The Future of Everything. I'm Russ Altman. I'm speaking with Professor Ali Boehm from Stanford University. In the last segment, Ali told us how her group figured out how to measure wastewater levels of COVID and other diseases in order to follow progress of the pandemic. They have amazing quantitative accuracy in the amount, and they can see changes in the virus as it evolves. In this segment, Ali will tell us how she got started with this project, and also she'll tell us how it's been trying to get other wastewater companies beyond her initial collaborators to adopt this technology. How did you ever think of doing this?
Well, when the pandemic was first starting in January or February of 2020, one of my colleagues from University of Michigan was here on sabbatical. Her name is Krista Wigington. And we thought that since the pandemic was starting in China, that we should try to get funding from NSF to do some work together on envelope viruses. And so we wrote a very small grant to NSF, and in it, we proposed to do a bunch of different stuff on envelope viruses, including looking for envelope viruses in wastewater, because that was something that was hadn't really been done before. And COVID
is indeed an enveloped virus?
That's right.
Okay.
Yes. And so these are viruses that have lipid envelopes around them, and they're not very common in feces. They're not very common for enteric viruses, which are the main types of viruses that are studied in wastewater. And so our program manager at NSF first said, no, I can't fund this because COVID is not, the novel coronavirus 19 is not relevant for the United States. And
Beautiful.
so, yeah, so we were watching the news and we saw that there was the first case in Santa Clara County, and we contacted the program manager and he agreed to fund us. So this was maybe $20,000 to do this project. And so as soon as we got our funding from NSF, Stanford University shut down and said, don't come back and leave. And we were like, wait.
Many labs, as you know, many labs were just closed down with very little warning.
Yeah. So we were able to stay and work in the lab and start acquiring these wastewater samples. And we, at that time, we had no idea if SARS-CoV-2 could even be detected in wastewater. Nobody had any idea if it could. And so once we established our methods in the lab, we established we could detect it in wastewater. We had some very promising results from our local wastewater treatment plant in Santa Clara County. We were able to get some philanthropic funding to continue our work and then eventually scale our work to provide daily measurements at 8 wastewater treatment plants in the Bay Area. And make that, we call that Project SCAN. And we partnered with a health science laboratory called Verily in South San Francisco as an industrial lab to process the samples very quickly within 24 hours. Huh. And now we received additional philanthropic support to expand beyond those 8 plants to 300 wastewater treatment plants across the entire country. And wow. So that's what we're working on now. So for me personally, it's been a pretty amazing journey to go from, you know, hacking in the lab with a bunch of grad students and postdocs to now running this like huge project that is going to have 300 wastewater treatment plants participating. And over such a short time, it's just been like 2 years. And I remember along the way at various times thinking, I can't do this. Like, this is not, I don't do this kind of thing, but I just put my head down and kept working really hard.
When you say you can't do it, do you mean that it didn't feel academic enough because it was too useful? I mean, what was the feeling that you had? I want to explore that a little bit. I can't do this because you obviously did do it.
There are many facets of that. There's the facet of I'm so exhausted from the pandemic and I just need a break and I can't do this, to I'm just an academic researcher. Like, I can't do something at scale like this.
Right.
I can't have people relying on me. And then there's also the freakouts occasionally that, like, is this what an academic should be doing?
Right.
And I've really come to realize that the only way that this is possible is exactly the way that it is, which I'm really happy about, is that I have a partner that is a commercial lab that can run the samples, because students and postdocs are not the right kind of people to run this scaled version of this project. It's just not fair to them. It's not fundamental research. It requires working after hours and on the weekend. So it has been a really interesting journey to go from, lab scale to like this large scale project.
So I want to get to the dissemination to the 300 or more groups, but I just have to ask, you must have benefited from this kind of open science idea. So I know enough about how PCR works that you needed to know the sequence of the genome of SARS. And so I'm assuming that came out and was publicly available, or at least available to you in a very rapid fashion. And I want to highlight that because that is not always the case in terms of sharing scientific data. And did you in fact benefit from these public disclosures of the genome?
Yeah, absolutely. And we continue to benefit from those as we design new assays for different variants. So the fact that the genomes of like newly discovered variants are discoverable and downloadable as soon as they're submitted and QA'd, that is a huge resource for us. And I think it's not the same way for all viruses. And so I hope that we get to the point where all that kind of data is easily accessible. And our project is also completely open data, open access, open protocols as well.
Great. Okay, so let's go to this now, dissemination and uptake by others. Has it been a group hug, where everybody says, thank you for doing this. We want to get on board. Here's our pipe, sample away? Or is it more complicated?
It's way more complicated. And I think the biggest challenge to wastewater-based epidemiology being uptaken and sustainable into the future, which I think it should, it's very important for pandemic and outbreak response. The biggest challenge is actually the coordination between the plant, public health, and the laboratory. And so it might be surprising to most people that wastewater treatment plants do not normally interact very closely with public health officials. So public health official may not even know how many wastewater treatment plants are in their neighborhood.
Are wastewater plants, if I can just ask, are they typically a government-run thing or a private industry?
Some of them are public, some of them are private. It just depends on the state. And so, I think from a wastewater treatment plant's perspective, at first, some of them were concerned about the topic we discussed earlier, like does detection of SARS-CoV-2 in our wastewater have implications on our permitting or our compliance with state laws and that kind of thing. And very quickly they learned that it does not. But even today with the whole polio thing, wastewater treatment plants are concerned that if polio were detected in their wastewater, that it would have impacts on the safety of their employees. So there's always those concerns that come up. And then for public health, as you alluded to earlier, we've had an amazing experience learning together in the state of California and in our local county how to use wastewater. And it is a group hug now. Like, I feel like we've come so far and we really like working together, but it's not the same in every state. And some states, as you mentioned earlier, they have a way of doing things and they don't necessarily want a new tool. And the other thing is that some public health departments actually do not think that data on wastewater should be public because they don't believe that the public is smart enough to understand what it means. And so they don't want to participate because they don't want their data to be public. And all data that is collected from wastewater, it's not protected data because it's not linked to an individual, and it's submitted to the CDC database where it becomes publicly available. So that's been sort of a roadblock in some places.
Yeah, so I can imagine that there start to be political considerations about, does this area X have certain diseases or not? And if the data is available, then people have to kind of explain it, and it could get complicated. But It sounds like in the long term, you are very bullish on the opportunities for wastewater surveillance combined with epidemiology being a huge light to shine for the benefit of public health.
Yeah, that's right. And just, we've thought about like, what are all the different uses for wastewater that we've seen and we think exist in the future. And we know it can be used for pandemic response. It can be used for like planning at hospitals for staffing, for example, and surge capacity. Mm-hmm. It can be used to inform some clinical decision-making, like testing, for example, the prescriptions of like therapeutics. And lastly, aside from like the sort of researchy epidemiology side, which it can also be used for, it could also potentially be used for vaccine development because you actually have the opportunity to sequence viral nucleic acids in wastewater and know exactly which strains are circulating. And if you can develop a vaccine quickly, and then you could administer it and you could protect the population. So that's like a pipe dream. I know There's a lot of pipe problems. Dream. Yeah. No pun intended.
Well, thank you so much. This teaches us all that every time we flush the toilet, we are potentially making a contribution to science. Thanks to Ali Boehm. That was the future of wastewater epidemiology. This is Russ Altman, and this has been The Future of Everything.